we need to talk a little bit about the activities associated with an action potential and how action potentials actually occur. The purpose of the nervous system is to send information along our favorite nervous system cells, the neurons, although I really like all of the neuroglia too, so I can't say they're my favorite, but they're the ones that get all the PR and all the press and have a lot of hoopla around them. So we're talking about action potentials because they are the way that we send information along a neuron and down an axon to either another neuron or one of the effectors of a neuron, which will be muscles and glands when we talk about the central nervous system. Let me repeat that. Effectors of the central nervous system are muscles and glands. And oh, by the way, this neuron is within the central nervous system because this cell right here is one of the central nervous system neuroglial cells and oligodendrocyte because it's making myelin. We need to talk about gated channels. So basically, channels are a way that we send information to another cell. And there are three types of gated channels: chemically gated, voltage gated, or mechanically gated. We have already talked about chemically gated channels because we saw chemically gated channels when we talked about the neuromuscular junction. The receptors on the motor end plate of a skeletal muscle fiber will bind with the neurotransmitter acetylcholine, specific for skeletal muscle contraction, which will cause a cascade of events, which in turn will trigger the release of calcium from the sarcoplasmic reticulum. So this guy is something you've been introduced to already. We also have voltage-gated channels. So these guys are going to open and close in response to what's known as change in the membrane potential, and we'll describe what a membrane potential is in a second or two. And last but not least, we have the mechanically gated channels, which are going to open in response to some sort of physical deformation of a receptor. So classic example of that is if you have pressure on a sensory receptor that is supposed to be picking up information about pressure, that gate will open and information will travel within the cell. So cell membrane potential, what is it? If you have a cell, a neuron in particular, at rest, there will be a very slight charge in terms of the voltage on either side of the cell membrane because of the amount of ions. You will remember that ions are charged particles, and there will be a different charge inside the cell than outside of the cell because of the difference in ions on either side of the cell membrane. And we're talking about tiny little differences. We're not talking about major differences. We're talking about millivolts, so a thousandth of a volt. And a voltage is basically just the measure of potential energy that is going to be generated by separate charges. And that's just a fancy way. All of this is a fancy way for saying there will be a very slight difference in charges on either side of the cell membrane because of where various ions are located. So that gives the cell what's known as a resting membrane potential. The cell is just hanging out and doing its thing. It hasn't been stimulated. And you're going to have a different distribution of ions on either side of the cell membrane, which we just mentioned. So potassium will be higher inside, and sodium will be higher outside the cell. And chlorine will also be higher outside the cell. Negatively charged proteins around ions are going to be in a slightly higher concentration inside the cell rather than outside the cell. The players that you need to know about when it comes to an action potential are sodium and potassium. So for the purposes of talking about an action potential, we talk about sodium and potassium and we sort of pretend the others don't exist, at least for the purposes of this course. Potassium will have a greater concentration inside the cell, and there will be a greater concentration of sodium outside the cell. Those are the two that you need to remember. We also have what's known as the sodium-potassium pump. So that's going to play a big role in helping maintain the appropriate concentration gradients of either of both sodium and potassium when we talk about an action potential. It's known as the sodium-potassium ATPase pump, but we just call it the sodium-potassium pump. It is a transport mechanism. It is an active transport mechanism. So gated channels are going to open or close depending on what is stimulating them, either electrical input or chemical factors. Again, we have a potential difference across the cell membrane. We know when we talk about a neuron, we're going to have greater concentration of sodium outside the cell and a greater concentration of potassium inside the cell, which means that there will be a potential difference or a charge difference of negative 70 millivolts. That's the number you need to remember. And as long as the cell is undisturbed and not stimulated, you will continue to have that slight change in charges. And the fancy word for saying that is that the membrane is polarized. And what do you need to know about this? You need to know this. The resting membrane potential of a neuron is negative 70 millivolts, and that can be disrupted or changed. It can either become more negative, which means it is hyperpolarized, or it can become less negative, i.e. a little bit more positive, moving towards zero, which is depolarized. When you talk about local potential changes, we know that neurons are going to respond or be stimulated by changes in the environment. That stimuli will affect the membrane potential in the region of the membrane that is exposed to that stimulus. The amount of change of that membrane potential will be directly proportional to the intensity of the stimulus. So if you have a little stimulus, there'll be a little change, which may not meet threshold and be big enough to cause an action potential. And if you have a big stimulus, you'll have a big change, and that is more likely to meet the threshold and cause an action potential. So let that sink in, and we'll continue on with more information about the action potential in the next section.